Hi, everyone. Welcome to the Tufts University USDA Human Nutrition Research Center on Aging's Monday Seminar Series. We're glad you're able to join us. Before introducing the speaker today, just as a bit of housekeeping, if you have questions, please use the Q&A tab in the Zoom and we will answer them at the end of the seminar. So today we're fortunate to have as a speaker Dr. Yong Xu. Dr. Xu is the Associate Director of another USDA Human Nutrition Research Center, the Children's Nutrition Research Center at the Baylor College of Medicine, where he is also a professor. His research focuses on understanding the neural signals that are critical for metabolism, eating behavior, and obesity. And today he is going to talk to us about the central regulations of feeding, cognition, and mood. Dr. Xu, thank you very much, and we look forward to your talk.
Thank you for the introduction. Also, I really appreciate this opportunity to share with you some of the work we do here in Houston, also in USDA Center. Actually, I consider myself as a basic neuroscientist with a physician's mind, largely because my clinical training when I was very young in China. Later, I switched my direction to pursue PhD training in basic neuroscience and has stayed as a basic neuroscientist ever since. However, I would say my early clinical training had profound impact on my research career because it always has driven me to do basic science that could make a difference in clinical practice, as you will see in my talk. Let's see how I advance my slides. Okay, so I started my independent research by looking at how the brain regulates feeding behavior as one of the metabolic behavior, with the goal to try to understand the physiology or etiology underneath some human problems such as obesity and the diabetes. As you can see from this hierarchy of demands, feeding behavior is one fundamental behavior essential for survival of animals as well as for humans. And therefore, it didn't take too long for me to realize that feeding behavior can be influenced by many other behaviors in the same chart, such as the thermoadaptive behavior, sleep, pain sensation, social behaviors, and so on and so forth. And these other behaviors can also be affected by feeding behavior or the metabolic signals. And so we often study. So the interplay between the metabolism and the behavior can be reciprocal. This is why after about 10 years of my independent research, my program has become so broad that now we are looking at how the brain regulates various behaviors as highlighted here. Which are relevant not only to the metabolic disease, but also for some neurobiological diseases such as Alzheimer's disease, autism, and so on and so forth. However, I still want to start from feeding behavior for this presentation today. So we focus a lot on this melanocortin neurons as you are seeing here. So so here, I don't know if you can see my mouse. So here you're looking at a coronal section of mouse brain. And then in the middle, this empty area, this is called third ventricle. On both sides of the third ventricle, this is at the bottom of the hypothalamus, you have this triangle structure called arcuate nucleus or ARH. Within ARH, there's at least two groups of neurons. These red neurons are called POMC neurons. They stand for pro-opium melanocortin. And then these green neurons, they are AGRP neurons, which stand for agouti-related peptide. So both AGRP neurons and the POMC neurons are considered melanocortin neurons because they both provide either a negative or positive melanocortin signaling in the brain. And the question is, why are we so obsessed about this melanocortin neurons? Because they are absolutely essential for feeding and body weight control. There's tons of evidence in the literature that can be used to support this statement. Here, I'll just show you one case, one example for each. For example, in this study that was published more than 10 years ago now, this group of investigators, they made a mouse model where the POMC neurons in the arcuate nucleus are genetically bladed or killed. So this is a group of mice that live without POMC neurons in the arcuate nucleus. And here they are measuring their daily food intake as shown by this by this red curve here over several days. Here, if you look at all the other curves, these are different kind of control animals. You see their food intake is about 3 to 4 grams. This is what the normal animals would eat, and normal mice would eat on a daily basis. However, if the mice live without POMC neurons in their arcuate nucleus, they're going to eat about 5 or 6 or even more, almost double this normal amount of food intake that a normal mice would eat. As you can imagine, if mice keep eating like this, very quickly they will become massively obese, as demonstrated here by this body weight curve. So this data indicates that POMC neurons are absolutely essential to prevent obesity, to prevent overeating. What about AGRP neurons? AGRP neurons are doing exactly the opposite. They drive animals to eat. And here, again, there's many evidence in the literature that you can use. And here I just show you something that were produced in the lab. So here we use a technology called optogenetics. Basically, this take advantage of this ion channel called channelrhodopsin, which is only sensitive to a blue light stimulation. So this would once they were exposed to blue light, and then this will lead to the opening of the channel, leads to the excitation of the cells that express this channelrhodopsin channel. So what you are seeing here is the 2 mice that we have engineered in a way that their AGRP neurons specifically express channelrhodopsin, and then you can see these fibers were surgically implanted towards the arcuate nucleus so that we can deliver the blue light pulses at any time of our choice. And for the first part of this movie, you see that we're not delivering any blue light. So this is serve as a baseline control. And again, this is in early morning at around 9 o'clock. So this is the time that normal animals would not eat, because mice eat most of their food during the dark cycle. So at this time of the day, they're not hungry, so they're not eating. That's what you're seeing. The animals were running around, they have food easily available for them, but they're not eating. Now, we start stimulating one of these mouse and still leave the other mouse as a control. As you can see that within a few seconds, you'll see this mouse start to eat, and only because we now activate AGRP neurons experimentally in this mouse. This is the same mouse that are not hungry about 2 minutes ago. Now, by stimulating AGRP neurons, we drive these animals to feel hunger, and then they start eating like they're hungry. Now, I'm going to switch to the last bit of the movie where we switch off the light, and then you see what will happen. So I'll give you still a few more seconds to enjoy the eating behavior of this mouse, because we're stimulating, stimulating, stimulating. Now we stop. The mouse almost immediately stopped eating. So I hope this kind of videos can convince you that the experimental activation of AGRP neurons can, even in a satiated mouse, can drive animals to feel hunger and therefore drive them to eat. So this diagram here is a very simplified version of mine to summarize what we understand about the melanocortin neurons and our feeding behavior. So typically when the animals are well fed, POMC neurons were firing at a higher frequency, and they will release their neuropeptide, and one of them is called alpha MSH. Alpha MSH actually is a gene product of the POMC gene. Alpha MSH is a neuropeptide which will act on the downstream neurons that express its receptor called melanocortin 4 receptor, or MC4 receptor. And this will lead to the excitation of MC4 receptor, and this ultimately will lead to the satiety signal. So this pathway largely explains when we have a big meal, we feel full, we stop eating. On the other hand, when the animals were fasted, POMC neuron will decrease their firing activity, and this adjacent AGRP neurons will start to increase their firing activity, and therefore release the neuropeptides or neurotransmitters within the AGRP neurons. And one of them is AGRP. What is really interesting is AGRP is an endogenous inhibitor of the same MC4 receptor. Therefore, will inhibit MC4 receptor and leads to the opposite signal, that's appetite signal. And then this pathway largely explains why when we are hungry, we like to eat. And then there's two things that I really want to emphasize about this melanocortin pathway. Number one is this pathway is not only important for feeding control in animals, but also in humans. Here, there's a list of the genes that has been shown and through genetic work in humans as well as in animals, has been shown that loss of function mutations of these genes can cause overeating, can cause obesity in humans. And then the mechanism underlying this phenomenon is because the impaired melanocortin signaling. And this includes the POMC gene itself, and then leptin receptor, and then leptin, so on and so forth. Here I highlight one gene called NCOA1, which encodes this protein called SSE1, steroid receptor co-activator 1. The reason I highlight this gene here is because this is based on our work in my lab. And then we published this about a few years ago, and then this actually result in a addition of this gene in the diagnostic panel for human obesity. And quickly, clinicians realize that there is actually many more patients who develop obesity and happen to also have the SSE1 gene deficiency. Right now, there are actually a clinical trial, phase 3 clinical trial, try to targeting this type of patients with a well-educated mechanism. And so because our work, we also show that the SSE1 deficiency actually impair the POMC gene expression, and therefore we reduce the tone of melanocortin signaling in the brain that leads to overeating and then obesity development. So a simple way to think about how to fix this issue is basically you have a problem upstream, and then maybe we can just simply bypass the upstream signal, directly activate the downstream signal, for example, by directly activating MC4R receptor. And then actually there is a selective agonist for MC4 receptor called the semaglutide, which has been proved by FDA to treat a number of genetic obese syndrome in patients. And then based on our work, this drug is currently under clinical trial to treat patients with SSC1 deficiency. So that's the point first point I want to make is from more clinical perspective is focusing on this melanocortin pathway leads to a lot of discoveries about not only feeding and obesity development in animals, but also in humans. And this can also guide our novel diagnostic methods as well as the development of treatment for certain obese syndrome. And then the other point I want to make is that from the perspective of biology, you can see that in this tiny little area of the arcuate nucleus, there's this interesting yin and yang system. You have AGIP neurons and POMC neurons. Their functions on feeding behavior are exactly opposite to each other. However, their firing activity are also oppositely correlated, but in a very nice way to meet the nutritional demands at different scenarios. So when you are hungry, you have AGIP neurons firing increase to drive feeding or food-seeking behavior. When you are full, the POMC neurons start to firing more. This will suppress appetite to reduce feeding behavior, to terminate feeding behavior. So a question in the field, at least to me, for my lab, in the last 10 years is how are the activities of AGIP and POMC neurons regulated to maintain the body weight balance in all kinds of situations? And then we keep asking these questions, and this led us to a number of new discoveries that may be very relevant for obesity. So today I will quickly go through a number of papers that we published, and some of them are still in sort of in the pipeline in the last 2 or 3 years. And then they're all sort of related to this melanocortin pathway. And then some of them actually also start to diverge to something that more related to cognition and mood, as you will see. Okay, so the first story I want to tell you is about this exercise mimetic that we discovered about 2 years ago called LAC-FEE. So this is in collaboration with Dr. Jonathan Long at Stanford University. Jonathan is a really talented molecular chemist, and then he's been interested in the molecular mechanism for the metabolic benefits of exercise. In this study, he started from the unbiased searching for metabolites in the serum in people who just experience intense running. So by doing this, he was able to identify consistently this metabolite always increase after a run. And then he was able to ID this metabolite called LAC-FEE. What is really interesting about the LAC-FEE is the same peak of LAC-FEE can also be observed in other species, including mice after running or a racehorse after a race. So this seems to be a very conserved metabolite in different species after the intense exercise. And then this is what lactate looks like. Basically, it's a very simple conjugate of two simple molecules. One is lactate, the other one is phenylalanine. And working together, the two groups were able to show that if we inject animals with lactate at a concentration that is similar to after a intense running, we're able to suppress animals' feeding behavior and the body weight. So indicating that lactate could be a very good target, or the metabolite itself, to be used to treat or to prevent obesity. So we published this paper about 2 years ago in Nature, and then we keep going to try to understand the mechanism, because people keep asking us, what is the mechanism for lactate to suppress feeding? So there's a long story, but I want to make it short. Basically, we eventually identify that the AGRP neurons is the cellular target for lactate to suppress feeding behavior. Here, just show you a piece of evidence. Here we use ex vivo in electrophysiology recording. So basically, we prepare the brain slices from animals that contain the arcuate nucleus, where the AGRP neurons are genetically enabled by fluorescence. So we can visualize this AGRP neurons. We can record from this identified AGRP neurons, and then we can puff a small dose of whatever you want, like feed or vehicle, and then we watch what is happening with AGRP firing activity. So shown here is example of one AGRP neuron we record at a current clamp mode, where you see this vertical spikes. You can maybe see from here because it's an S-dense. So each vertical spike represent a action potential. If you see a very dense spikes, that means the firing frequency of action potential is very high, which is basically you are seeing here. The baseline is had a high firing frequency. If you treat this with a vehicle, you don't really see that change, indicating this is a good, very stable baseline. Then if we treat the same cell with a small dose of lactate, very quickly you'll see this firing frequency decrease. Eventually this become a silent AGRP neurons. So this indicates that lactate can efficiently suppress the firing activity of AGRP neurons. Of course, this is done in vitro. We want to confirm this in vivo. So we use a technology called fiber photometry. Fiber photometry takes advantage of this genetically engineered protein called GCAM. Basically, it's a calcium sensor protein, which can detect the amount of calcium inside the cells and then give away the green fluorescence proportional to the amount of calcium. So you can consider that as a calcium imaging if you're doing that, but this is in vivo. So because of this protein, we can engineer this into a viral vector in a created-dependent manner. We can deliver this into the AGRP Cree animals only in the arcuate nucleus. Therefore, only the AGRP neurons will express this calcium sensor. And at the same time, we can implant a optic fiber, which in this time is a detector of the green fluorescent intensity. And then we can measure that in freely moving animals, and use that as an estimate of AGRP neuron firing activity in animals when they're freely moving. So when we do this, we can also give animals injections. Shown here, the red curve is IP injection of lactate, which we show that it does significantly reduce the firing activity of AGRP neurons compared to number of other control injections. So this confirmed that even in vivo, lactate can suppress AGRP firing activity. Okay, so now the next question is whether this inhibition of AGRP neuron firing activity is a critical event to mediate the effects of lactate to suppress feeding. This seems to make sense, right? Because I show you when the AGRP neurons is artificially activated, you will drive animals to eat. So if you inhibit AGRP neurons, you probably expect the opposite response. So they will suppress their feeding behavior. But how do you prove that? So here we use a technology called DREAD, which stands for designer's receptor exclusively activated by designer's drug. I hope this make a little bit more sense when I read this out, but I'll explain this. So you can consider that you have a designer receptor, basically is a mutated G protein couple receptor, right? So the receptor is mutated in a way that it no longer responds to any of the endogenous ligand in the body. Instead, it only responds to a synthesized compound called designer's drug. And this compound actually has a name called CNO. So now, if you can deliver this expression of this DREAD receptor or design this receptor only in AGRP neurons or whatever neurons you want, you can use CNO injection at any time of your choice to specifically activate this receptor. And then you can make it several different versions. One of the version of this receptor is to couple to GQ. So usually the one leads to the activation of the neuron. So basically, that's what we did. So we delivered the drug receptor coupled to GQ in the AGRP neurons in these animals, again, using viral approach. And then we can inject these animals with signal at any time of our choice to artificially or experimentally activate the AGRP neurons in these animals. Shown here is the result. So here at the top, this is still in the control situation where the AGRP neurons are not experimentally manipulated. In this case, if we give animals injections of vehicle versus LAC-V, again, we can recapitulate what we produce this data. LAC-V can significantly suppress animals' feeding behavior. However, if we artificially drive AGRP neurons to increase their firing activity, in this case, LAC-V no longer works to suppress feeding. So this data indicates that LAC-V reduces feeding by inhibiting AGRP neurons. Then the next question is how LAC-V can suppress AGRP neuron firing activity. I will directly jump to the data that to answer this question. We actually observed that LAC-V can induce the KATP channel activity, increase the KATP channel opening. So KATP channel stands for the ATP sensitive potassium channel. So basically it's a potassium channel. When it is activated, the channel open up, the potassium will go from inside the cell to the outside the cell. And then because potassium is a positive ion, so this actually what leads to a inhibition of the cell, of the neuron. So shown here is basically a voltage clamp recording in AGRP neurons, and then we use a protocol that can measure this potassium hour current. As you can see that the vehicle treatment does not change this current, but LAC-V can efficiently increase this current. And then further we'll confirm this to be a KATP current because the KATP channel inhibitor tobutamide can specifically abolish this response of LAC-V. So basically this is a quantification of the same data, indicating that LAC-V can increase KATP current in AGRP neurons. And then that leads to the next question is whether, before I go to the next slide, and then we also went further to show that whether this opening of a KATP channel is physiologically relevant to mediate the effects of LAC-V on feeding behavior. I'm not going to show you the data, but we do have both genetic and pharmacological data to indicate that it is the case. The LAC-V can suppress feeding because its effect on KATP channel in AGRP neurons. Then the next question is how LAC-V can open up the KATP current in AGRP neurons. Here I show you a paradigm from this paper where they showed one of the mechanisms where the KATP current can be regulated in neurons. So as this name implies, this is an ATP sensitive potassium channel. One of the pathways to regulate ATP, the potassium channel is through the intracellular ATP levels. So when the intracellular ATP level is high, the KATP current will be reduced or closed. And when the ATP level is low, KATP channel will open up. This will lead to increase KATP current. Okay, then the question becomes how LAC-V may influence the intracellular ATP level in AGRP neurons. Now, here they shows one of the pathways that can influence intracellular ATP level in neurons. We know that neurons can take up a lot of lactate, and the lactate can interact with this protein or enzyme called lactate dehydrogenase or LDH. And the LDH will take lactate as a substrate and convert it into pyruvate. Pyruvate will then go into the mitochondria and go through TCA cycle, the mitochondrial respiratory chain reaction, and ultimately leads to the production of ATP. Now, let me remind you, what is the molecule of LAC-V? That is a simple conjugate of two other simple molecules. One is phenylalanine, the other one is lactate. So based on this structure, this prompts us to think about one possibility, that is LAC-V may be able to use the lactate half to interact with the same enzyme protein LDH, but function as an inhibitor, therefore can prevent lactate interaction with LDH, therefore can block this pathway. So we did a number of assays. One is to use the thermal shift assay to confirm that LAC-V does interact with the LDH protein, actually at a very high affinity, higher than lactate itself, which I'm not going to show you that data for. And then the other thing is at the functional level, what happened to the LDH enzyme activity? And then there is activity. There is LDH activity assay that you can use, and then we can show that LAC-V can dose-dependently suppress the LDH enzyme activity. So based on this, we think what may be happening here is when there is high level of LAC-V in the system, the LAC-V can interact with LDH and therefore occupy the LDH protein and therefore prevent the binding of lactate, the normal substrate, with LDH, and therefore inhibit this pathway leads to the inhibition of ATP production. And then this will open up the ATP sensitive channel, potassium channel, and leads to the inhibition of AGIP neurons. At a physiological or behavior level, this will lead to a suppression of food intake. All right. So just to summarize this part of my talk, we show that LAC-V is a exercise mimetic conserved in multiple species, including humans. And the LAC-V can suppress food intake in the body weight through inhibiting AGIP neurons. And then we found that LDH is at least one molecular target for LAC-V. And LAC-V and its analog has the potential to treat obesity and the metabolic disorders. Actually, we already patented this, and then we're in the negotiation with certain vendors, and hopefully this will come to the market very soon. And also I want to emphasize that because exercise actually is known to have a lot of other benefits, including cardiovascular benefits and the cancer benefits, and also cognitive benefits. So the lab also is looking into all these other possibilities, whether LAC-V as a exercise mimetic could produce some of this, mediate some of these benefits induced by exercise. And we do have some quite promising data in all these aspects. All right, so let me switch my gear to the second story where we focus on a G protein coupled receptor called serotonin 2C receptor and how this may be related to human obesity and cognition. So this is in collaboration with Dr. Sadaf Faruqi, who's a professor at the University of Cambridge. Sadaf is pretty well known in the field of obesity because of her contribution to the understanding of the genetic basis for human obesity. So basically, she has accumulated a big cohort of children in UK mostly with a very severe early onset obesity, and she has done a genetic workup in these patients to figure out what may go wrong in their genome, and then identify a number of variants, a lot of genetic variants that may be associated with childhood obesity. And then one of this gene is serotonin 2C receptor gene. And then shown here is the protein structure or motif of the serotonin 2C receptors. You can see this is a 7 transmembrane domain. So this is a G protein coupled receptor. And then this red circle here represents the amino acid that's been found to be mutated. And then this is identified from her big cohort of children with severe early onset obesity. So we collaborate. This is actually the second project we collaborate together, and then we use a very similar strategy. So after she identified this in humans, and then we took these genetic mutations. We first analyzed them at the cellular level to answer the question whether this mutation causes loss of function or gain of function, or they don't have a functional consequence. So basically, we took all these 13 mutations in serotonin 2C receptor. We performed a series cellular characterization, and we basically identify all of them show one or more of loss of function in terms of serotonin 2C receptor signaling. And then we pick this one, F327L mutation. This shows the most severe loss of function mutation. And then we pick this one and then make this into a mouse model. So we basically made a mouse which carry the same mutation that human has, and that is associated with human obesity. The question is whether mouse carrying the same mutation also recapitulate the human phenotype. The answer is yes. So shown here is regardless whether there's male mice or female mice, as long as they carry this mutation, they develop obesity compared to their leader-mate controls. And then they gain more weight because they eat more. This is measured through the metabolic cages where we can measure their minute-to-minute food intake over several days, and then we show that they are hyperphagic. So basically this data we capture the human phenotype. They eat more, they develop obesity, and then this is very significant because the combination of our mouse genetic data and the human genetic data basically provide perhaps the most definitive evidence to support the statement that the loss of function mutation of serotonin 2C receptor gene can cause human obesity. And then we suggest that this gene should also be added to the diagnostic gene panel for human obesity. Actually, this did happen after our paper was published. And so, but this is not where we're going to end, because now you can basically help and provide the genetic diagnosis for a group of obesity individuals with obesity, but can you help more? Can you help treat them, right? So that is the ultimate question, ultimate goal for our basic research. So that has led us to keep digging the mechanism, the pathophysiology. If we can understand better why this genetic mutation cause overeating, cause obesity development, maybe we can guide the new treatment for at least these individuals. And again, I just show you a few piece of key results. Here we went back to electrophysiology again, but this time we record from identified POMC neurons. Remember the POMC neurons, and their function is to suppress feeding. If they have impaired function, the animals will eat more, they will gain massive obesity, right? Remember that? So now we're recording from identified POMC neurons, and we treat them with this compound called lorcaserin. Lorcaserin is a selective serotonin 2C receptor agonist. As you can see here, the treatment of lorcaserin can actually increase the firing frequency of the wild-type POMC neurons. This makes sense, right? So activation of POMC neurons needs to increase melanocortin signaling in the brain. This will therefore suppress the appetite and suppress feeding. But what will happen if the animals carry this serotonin 2C receptor mutation? You record from POMC neuron, you found this activation of POMC neuron induced by lorcaserin is significantly attenuated, as demonstrated here. So this leads to the possibility maybe this impaired melanocortin signaling induced by the serotonin 2C receptor mutation is the culprit, is the reason that leads to overeating and obesity. To try to prove that, we did this experiment here. I'm not going to go into the technical detail and just directly tell you the results. So here, if you look at the gray curve, this is a group of wild type mice. You're looking at their normal body weight curve. And then the red curve here is a group of mice that are carrying this serotonin 2C receptor mutation. Again, we recapitulate the human phenotype, they gain more weight. And then more importantly is this blue curve. So this is a group of mice that also carry the serotonin 2C receptor mutation in all their cells. But at the same time, in their POMC neurons, and only in their POMC neurons, we put back a wild-type copy of serotonin 2C receptor. And our question is whether by doing this, can we can rescue the obese development? And the answer is yes. By putting the serotonin 2C receptor only in POMC neurons at this background of serotonin 2C receptor mutation in all cells, we can prevent obese development. So that indicates these POMC neurons are important to mediate the effects of serotonin 2C receptor action to suppress feeding. It also indicates that the obesity development, overeating in this mutant is likely due to the impaired melanocortin pathway. Again, we use a very simple logic. If that's the case, let's bypass the upstream signal that is being impaired. Let's go directly to enhance the downstream signal, which is MC4 receptor agonist. And then we suggest that this agonist can also be used to effectively treat individuals that develop obesity and happen to have the serotonin 2C receptor mutation. So that's our conclusion here. So while we're doing this experiments, we also noticed some very obvious phenotype from the serotonin 2C receptor mutations. That is, we found these animals are not that smart compared to their wild type leader mates. So then we started to formally test the cognition or learning memory functions of these animals. One of the assays we use is called the radial arm water maze. So I'm sure that in the aging center, many of our colleagues there are using similar cognitive assays just to simply go through this in case somebody do not usually do this. So basically, we have this different 6 arms, and then they're filled with water, and then the animals were put in the water, and the animals don't like to be in the water. They're trying to find the escape, right? So we always put one platform in one of the arms, and then so the animals, once they find this, they will find escape. They will stay there. So if you train animals a number of times, hopefully they will remember. They will learn this, and then they will remember this. Next time when you do the test, they will quickly find the platform in the same location. So that's what happened with the wild type mice. You see, they quickly, and this is showing the latency of finding the platform. You see, they take a very little time for them to find the platform. However, if you look at the mutant animals, this is the same mice that have the serotonin 2C receptor mutation. We found that they take much longer for them to find the platform. And actually, you see the 60 seconds, this is when we have to rescue. The experiment was designed for only 60 seconds. If the mouse cannot find the platform, we just stop it. And so these 2 mice actually didn't find the platform at the end. And this assay, as well as the field conditioning assay, which I'm not going to show you, both confirm that these mutant animals do have impaired memory. So we told this discovery in mice to our collaborator, Furuki, who has the patients. We told her that maybe you should look at formally check their cognitive function in patients. So I have to make a point here. So in collaborating with our clinicians, a lot of time it's the basic neuroscience, basic side. We are enjoying the inputs for our clinical colleagues, and they made observations in clinical setting and then guide us to look for similar phenotypes in animals and guide us to look for mechanisms. But occasionally we also have this reciprocal, the other way around of a relationship. This is one such example. We made a observation in mice, and then we told them, so maybe you should take a look at it. So that's what the staff did. So she collect a few patients who carry the serotonin 2C receptor. This is young adults, but carry this serotonin 2C receptor mutation, and use this questionnaire to check their learning and memory. And I did find that these individuals have actually quite severe impairment in their memory. This is when they're very, very young. Okay, so this gives us confidence that we are looking at something that is relevant, not only in mice, but also in humans. So then the question again is, what is the mechanism? How serotonin 2C receptor is important for learning and memory? So for this, we naturally turn our attention from the hypothalamus to the hippocampus, because hippocampus is well known as where the memory should be stored. And then within the hippocampus, we know the serotonin 2C receptor is more abundantly expressed in this subdivision called the ventral CA1, as shown here. So we start to look at ventral CA1, where the serotonin 2C receptor is highly expressed. And then one experiment we did is let's knock out the serotonin 2C receptor only in ventral CA1. Here we use the serotonin 2C receptor flox animals. Wild type littermates was used as control. Both of this group of animals receive the AAV pre-injections in both sides of ventral CA1. So in this flox animals, the 2C receptor will be selectively deleted only in the ventral CA1, where the wild type do not have that deletion or serve as control. This is RNA scope showing the messenger RNA of serotonin 2C receptor in the ventral CA1 in control animals. And then you see in the knockout animals, we successfully delete most of the serotonin 2C receptor there. And then we use the functional assay. We show that, yes, when you specifically knock out serotonin 2C receptor only in the ventral CA1, you also cause this memory decline. And then the question becomes, okay, so we have the receptor in the ventral CA1 that has a function that almost dictate that this region must receive the ligand somewhere in the brain, right? The serotonin 2C receptor's ligand is serotonin, which is a neurotransmitter. So we know that most of the serotonin 2C serotonin is produced by the serotonin neurons in the high brain, particularly in this region called the dorsal raphe nucleus. I'm sorry, dorsal raphe nucleus. And so in this experiment, we took advantage of this mouse model called TPH2 flox animal. TPH2 is the key enzyme for the production of serotonin in the neurons. So if you knock out TPH2, you basically will deplete the serotonin production from that neuron. So what happened here is we took TPH2 flox animal here, And then we inject the retrograde AAV Cree into the downstream target, the ventral CA1. So this virus will be picked up by the terminals in the ventral CA1 and go upstream to whatever neurons that project to the ventral CA1. This would include some of the serotonin neurons in the dorsal raphe nucleus. And then the Cree will induce the recombination of the Flox allele. Therefore, we move or delete the TPH2. And therefore, these serotonin neurons cannot produce serotonin anymore. And remember, this is the serotonin neurons, only the subset of serotonin neurons that project the ventral CA1, not all the serotonin neurons. So we validate this. We show that the normal situation, you have the TPH2 expression in the dorsal raphe nucleus. And then in this particular knockout, we found some of the serotonin neurons lose their expression of TPH2, therefore they cannot produce serotonin. And then the result is very similar. The animals had hard time finding the platform in the water maze. And then why is it any relevant in humans other than that, a few individuals may carry the serotonin receptor, 2C receptor mutation, they have a memory decline. So we keep thinking whether this knowledge that we learn from this few individuals as well as from animals can be applied to a more bigger problem in human society, such as the Alzheimer's disease. And I don't think, at least so far, there is no evidence to indicate that Alzheimer is associated with mutation of serotonin 2C receptor per se. I don't have any evidence. Many people ask me that, but we don't have evidence for that. I don't see any evidence for that. However, in the literature, there is a lot of reports about this phenomenon called serotonin denervation in human AD brains. So shown here is one example. This is published by others. You see this is from human brain, a normal brain. You see, again, this is serotonin neurons in the dorsal raphe nucleus. The human brain looks slightly different as the mouse brain in this region, but you can still see this kind of jellyfish, you would say, jellyfish structure in the dorsal raphe. Then this is the same region in an AD patient's brain. And then you can appreciate that the number of serotonin neurons is reduced. There are also reports about not only the numbers of serotonin neurons cell body reduced, also there's projections and fibers coming from serotonin neurons could be damaged in the AD brains. So they may not be a deficit in the receptor of serotonin 2C receptor. However, the ligand may be impaired in the serotonin in the AD patients. So this prompts us to think about whether what we learn from the 2C mouse and patients with 2C receptor mutation can be applied to the AD patients. So here we use a mouse, a AD mouse model called the APP-NLGF. So this is a knocking mouse model that have 3 different mutations in APP gene, which is being identified in humans. And then this model is known because it's very aggressive. So we think as early as 6 months of age, these animals will develop, tons of very abundant deep deposition of A beta and beta amyloid throughout the brain as demonstrated here. Also at 6 months of age, you will be able to start to detect some memory deficits. And so that's why we choose 6 months of age of these animals. And then what we're looking at is the serotonin neuron cell bodies and the serotonin neural fibers projecting to the ventral CA1. I'm not showing you the serotonin neuron cell bodies in the dorsal raphe because the difference is very subtle. However, in the ventral CA1, as you can see, the serotonin fibers, the projections, as demonstrated by these red fibers, they're significantly impaired in this AD model. So this is kind of similar to what I show you from the human brains. So this indicates that in the AD brain, at the early stage of disease development, because this is the time when we start to observe behavior deficits, there is already a very profound decrease of serotonin innervations in the ventral CA1 region. And we know that in this region, it's the serotonin 2C receptor that is functional, that is very important for mediating the memory. So that leads to our next question. Again, can we bypass this? Can we directly stimulate the downstream receptor to improve the memory function? The answer is yes. So here we use 6 months old animals, wild type control versus APP-NLGF animals. They receive either saline injection or lorcaserin injection. Again, lorcaserin is the selective agonist for serotonin 2C receptor. And then we're looking at their behavior in the water maze assay. So if you just look at the Y bars here, this is wild type, receive saline, or saline-treated AD mouse model. And as reported by others, we also recaptured this as early as 6 months of age. This AD mouse model start to show significant deficits in memory behavior. And then if the animals were treated with serotonin 2C receptor agonist, we can improve this behavior, indicating that the serotonin 2C receptor agonist can improve cognition in this AD mouse model. So to summarize this part of my talk, so loss of function mutation in serotonin 2C receptor cause obesity and the cognitive decline. And then the 2C receptor gene has been included in the genetic diagnostic panel for human obesity, and the melanocortin analog, the cell mannitide, can be used to treat obesity with 2C receptor deficiency. And then we also found that the 2C receptor, the serotonin to ventral CA1 circuit maintains normal cognition. This circuit is impaired in a AD mouse model, and the serotonin 2C receptor agonists, such as lorcaserin, can improve cognition in this AD mouse model. All right, so for the last part of my talk, I'm going to switch my gear again, talking about this different gene called TRPC5 and how this may regulate feeding and the maternal health. Again, this is in collaboration with Dr. Salah Faruqi. We're fortunate to be able to publish this work early this year in Cell. So the paper basically described our discovery about the TRPC5 mutation that she identified in humans, and then we further characterized using animal models, and then found that this gene actually play a very important role in many of the human diseases, including obesity and hyperphagia, and also postpartum depression, anxiety, autism, and then maladaptive behavior, as well as the sleep disruptions. I'm not going to show you all this data, just to show you a few interesting stuff. So the story actually started from this human characterization. Again, so Sadaf has 2 boys who showed a very early onset severe obesity, as demonstrated here. So I want to emphasize that this gray area is the normal weight of boys, 5% to 95%. Luckily, all my children are within this range. However, if you see these 2 boys, they're totally out of the chart. It's very, very severe obesity. And then both these boys had mutations in the serotonin 2C receptor genes. And then also, before I move on, so also the Sadaf also characterized the her big cohort and identified 7 other point mutations in the TRPC5 gene, which is associated with early onset severe obesity. And then we took a very similar strategy. We first characterized all these point mutations, make sure they're loss function mutation, and then we picked one mutation that is most severe and made into a mouse. And then we found the mouse recapitulate all the human phenotype. This is all reported in this paper. So I'm not going to go through all that, because I think by this time you're already bored by looking at feeding and the body weight curves, right? But I want to show you perhaps 2 additional things that are quite interesting to me. So first, let me read the description of one of this boy. The other boy is actually very similar. So the boy was hyperphagic from infancy. In childhood, display intense food-seeking behavior, stealing food, hoarding, and hiding it for later consumption. His parents went to extreme lengths to hide food in the tumble dryer, such as in the tumble dryers. So the reason that I highlight hoarding is because when the first time Sadaf told me about this through a Zoom meeting, of course, verbally, I had to look up what hoarding means. I didn't know what hoarding means. Of course, I know the concept of hoarding. You know, living in Houston, whenever we have a broadcast that a certain hurricane is about to hit us, everybody rushes to the Costco or other supermarket to pretty much buy everything possible. Food, water, even toilet papers, for reasons that I don't quite understand. So that's hoarding. I know the concept of hoarding. However, as a Chinese man, I never really used this English word in my daily life. So when the first time I actually heard from Sadaf, and now I have to look it up. What does it mean? And now we're like, oh, this is what she meant. And here I want to use a quote from my dear daughter. At the time, she was a 6th grader, and then she was writing an English essay to about OCD, obsessive and compulsive disorder. And then within this symptom, this disorder, there is actually one category is called hoarding disorder. So the concept, the compulsive needs to keep stuff can be a way of managing upsetting and then intrusive thoughts like concerns over cleanliness, health, or other obsessions or things that they need. So that's what the hoarding really means. But this really struck us because, first of all, even Saraf has been studying a lot of individuals with severe obesity due to other genetic mutations such as POMC, leptin receptor, MC4 receptor, so on and so forth. For most of these genetic obese syndromes, they never saw hoarding behavior. This is the first time, or very few occasions, where they observed such behavior in people. And two, if you dive into the animal literature, there's actually very little and literature about the neurobiological basis for hoarding behavior. So we thought that maybe this is an interesting observation that will lead to some discovery that could be even more interesting. So we decided to dive into this a little bit more to see whether we can measure hoarding in our mice. So it's not an easy thing. So because if you animals living in a home cage, they have no need to hoard, and there's no place to hoard, right? So my postdoc fellow had to DIY this device so that we can measure hoarding behavior. So basically, this is a mouse cage, home cage, and then she made a hole on one side and then connected with the steel tubing, and then basically she put a lot of food pellets at one end of the this tubing, and then let animals just observe overnight. Next morning, she come and measure how much food has been relocated to the home cage area, and then we use that as an estimate of hoarding behavior. So shown here is one piece of results, and when we first do this in the room temperature, so room temperature for mice, we set up at 24 degree. Basically, it's our own room temperature. And at this condition, we found that the mutant animals always show a bit of increase in hoarding behavior, but really never get significant. Then we thought, let's switch animals to a different room where the temperature can be set at a 28 degree. This is closer to thermal neutrality for mice. And then in this case, what we noticed, first of all, is the wild-type mice actually significantly reduce their hoarding behavior, which makes sense because when at the room temperature 24 degrees, this is actually a mild cold stress for animals. They have to spend more energy to defend their body temperature. Therefore, they may have more need to maintain food availability. So they like to hoard more food. But when they are living in thermal neutrality, they don't have that kind of metabolic needs. So the hoarding behavior will reduce. That's what we observe here. And then what is important here is this TRPC5 mutation mice, they do not reduce their hoarding behavior. If anything, they seem to increase a little bit. And then they showed a significant increase compared to the wild type animals in the same situation. So this data indicates two things. One, hoarding behavior in mice is also a temperature sensitive issue. And two is TRPC5 play a physiologically important role in regulating this behavior. And then one thing I should also emphasize that TRPC5 and other TRPC channels were all implicated as temperature sensors. So this may have some mechanistic link there for this innate behavior, which really interested us. And now this is not the end of the story. We're still trying to figure out what's going on with the hoarding behavior in the context of TRPC5 mutation. All right, another thing that I think is worth mentioning is about postpartum depression. So this is, again, the families of the two boys. So these are the two boys that I show you the body weight curve for. And then you see they're very young, very, very obese in terms of the BMI or the BMI deviations. And then their siblings who also carry the same mutation are also very obese. All these children actually inherited the same mutation from their moms, who also are mutated and then shows obesity. At the same time, both these moms experience very severe postpartum depression during every single of their pregnancies. So this become very significant in the clinical setting. When the staff interviewed this mom, she realized, oh, this is real. This is not just like a baby blue. It's very severe postpartum depression. However, this is significant in clinical setting, but it's not clinical significant in terms of statistic setting or scientific setting because she only has 2 patients. She told me about this, and then I said, no problem. We have mice, and then we can characterize this in mice as many as we needed, right? So, okay, that's why I start reading about postpartum depression. Up to this point, I never thought about that I will be doing research related to postpartum depression, but the clinical observations led us to this point. So this is some homework that I did. So apparently, it's a big problem. About 10 to 30% of mothers will suffer postpartum depression. And believe it or not, about 10% of fathers also claim that they have postpartum depression. And in terms of symptoms, of course, depression should have depression symptoms. I'm not going to read all the symptoms. You know that, right? But what is important is this depression symptom is specific during the postpartum period. It's not at the regular period. If it happens at the regular period, that's major depressive disorder. It's not postpartum depression, right? So that is very important. The other thing I think it's often overlooked, but also very important, is a very important characteristic of patients with postpartum depression is they do not like to take care of their babies. So they have impaired maternal behavior. That's why when we characterize our female animals, we look at their maternal behavior. So shown here is a movie. So we use a very classic maternal assay. So it's called a pup retrieval assay. So you basically put the pups to the opposite corner of the nest, and the wild type moms on the left and the mutant mom on the right. So you just quantify how quickly, how efficiently they can retrieve their pups. So you see this wild type mom is very efficient, very good. By this time, she's almost done. And then you see this mutant mom is just slower and less efficient. And then the other thing that you may notice that she doesn't care about nest at all. Very quickly, she disrupted the nest, whereas the wild type mom maintained the nest very well. So now I'm playing at 10x. So I hope that makes the point. The mutant TRPC5 mutation impair the maternal behavior in female moms. Okay, then postpartum depression has to have the depression during the postpartum period, right? But you cannot ask mice whether you are depressed, right? But there are a few assays that you can use to estimate the depression-like phenotype. One of them is called full swimming test. So basically, you just put animals in the big jug of water. Again, animals don't like to be in the water. They will struggle. They'll try to get out. But at a certain point, they will realize it's hopeless. It cannot get out. So that's when they give up, they will spend time immobile. They're not moving. So basically, we just count within the same amount of total observation time, we count how much time they spend immobile. The more time they spend immobile, they are more likely to be depressed. So shown here is the results. The mutant moms show significantly more time as immobile, indicating they have more increased depression-like phenotype. Another assay we use is the sucrose preference test. This is easy. Basically, we just give animals free choice of two bottles. One's water, one's sucrose, just like humans crave for sweet stuff. So this wild type animals drink about 8 to 90% of their water or liquid as sucrose, but you see the mutant moms show significantly reduced sucrose preference. So this indicates that they have a condition called anhedonia, which is another sign of postpartum depression. Now, by this time, you may ask me, do you observe this after weaning, after they give a baby? What about when they're virgins? So we perform these assays when they're virgins, same mutation, but there is no phenotype whatsoever. So the depression-like phenotype is highly specific during the postpartum period, which is really, really striking. And then we also look for the mechanism that may explain this. We found TRPC5 is highly expressed by this oxytocin neurons in the PVH, paraventricular nucleus of hypothalamus. And oxytocin, we know, is one of the nerve hormones that's important for not only maternal behavior and also mood regulation. So we did two different experiments. One experiment is we knock out TRPC5 only in oxytocin neurons. And this basically recapitulate most of the phenotype. They eat more, develop obesity. They also have problem retrieving pups. They are more depressed during the postpartum period. We also did the opposite experiment. We start from the mutant animals. They have the TRPC5 mutation in all cells, and then we put the wild type TRPC5 back to the oxytocin neurons, and then we can improve all this phenotype. So this data indicates that oxytocin neurons mediate TRPC5 effect on these behaviors, the postpartum mental health, as well as the maternal behavior, as well as feeding behavior. So what about the fatherhood?
Dr. Xu,
Yep.
We're very close to time, and we do have a couple questions. So I'm wondering if we can wrap this up.
I just have two slides. Yeah. So I told you that about 10% of fathers claim that they have postpartum depression. You may not believe, right? So I show you an example that belongs to the other 90%. This is actually myself about 2 years ago when we had our third baby. You see, I'm certainly not depressed, right? And then also I'm very skilled at what I'm doing. But I can tell you that for mice, this is not something that everybody is born with. If you took a cohort of male mice, virgin male mice, you give them several pups, randomly, these mice will show one of the three phenotypes. Many of them will attack pups. Also, many of them will ignore. Only a few of them will retrieve pups like good daddy, but they're a virgin. But what is really interesting is if you let these male mice to mate, to become biological fathers of others, and then you repeat this assay, again, you give them non-biological pups, just to be fair. And then you found all of them all of a sudden become super daddies. They start to retrieve the pups. So for males, this paternal behavior is something that is based on experience. That's why we perform this assay in two different stages, when the males are virgin or when they become fathers. Oh. So this is the last slide I want to show you because this is so interesting. So this is when there are virgins, wild type on the left, mutants on the right. So you can see that they're equally bad compared to the moms that I showed you earlier. I'm playing at 10x. So now I'm going to switch to the second half of the movie. And then I want you to focus your attention on the left-hand side of this for the first few seconds, because if you missed it, you're going to miss the whole thing. Again, the left-hand side is wild type fathers. Oh. Yeah, so focus on this left-hand side for the first few seconds. So you see this father finish it already within a few seconds, but the mutant fathers were still working on the pups. So they definitely have impaired paternal behavior. Right, so I'm gonna also show that they have depressed like phenotype when they become fathers, but they are normal when they're virgins. So I'm just gonna skip the conclusions. That's all I want to show you. This is the acknowledgement slides. This is a group who and I'll highlight the people who actually contribute to the work that I talk about today, and also our funding. And I'll highlight the ones that contribute that support the talk that the studies that I mentioned today. These are our collaborators throughout the world. And then sorry for the delay, and then I'll take your questions if you have any.
Thank you. So there are a few questions in the Q&A, and if anyone has more, please go ahead. But the first is from Jasper Weinberg, asks, what effects does LAC-Phi have in other cell types that have ATP-gated ion channels?
And we haven't tested a whole lot of other cell types. We did look at POMC neurons, for example. We do not find that it has effect on KATP channel in POMC neurons. And I think that's because the relative abundance of several things. First of all, LAC-Phi itself, also lactate, as well as the enzyme LDH. And so somebody already showed that using single cell RNA-seq approach, that POMC neurons have higher level of LDH compared to AGIP neurons. So I think the reason that may explain our observations, because you really need a whole lot more LAC-Phi in order to occupy the high level of LDH in POMC neurons in order to produce an effect that is detectable, whereas in AGIP neurons, because the LDH level is relatively lower, and then at the physiological concentration of LAC-Phi, we were able to observe that response. I hope that addressed your question.
Thank you. The second question is from Andrew Tarr. He says, thank you for your presentation. Have you looked if it is specifically acquisition or memory? And have you confirmed the cognitive deficit in other behavioral tests other than the radial water maze?
And we don't think it is during the acquisition phase, because we also have data during the learning phase, and we do not see problems. We think it is memory, and then we did look at it in the field conditioning assay, and then also the novel objective assay. So the the field conditioning assay shows very consistent result as the water maze. The novel objective assay shows somewhat a weaker response.
Thank you. The next question is from Zhangdong Wang. He says, great work and presentation. Thank you. Eating behavior is not only determined by energy state, but also our mind. For example, healthy and lean people and patients with obesity may reduce their food size when considering eating for health or opposite, such as by depression. I was wondering if the mindset could affect activities of the ARP or POMC neurons as potential mechanisms for eating behavior.
Absolutely, absolutely. So the feeding behavior is influenced by many factors. Most studied is homeostatic needs, of course, and also there's hedonic needs or palatability of the food, and also there's increasing evidence showing that the mood or the social context can also influence how we eat and how much we eat. So you're absolutely right. And also we actually published a number of years ago, we published that the psychological stress induced by restraint, for example, can regulate POMC neuron activity and therefore influence their body weight and feeding behavior. So and then other groups also shows similar stress models can influence AGIP neurons and influence their feeding behavior. So that's absolutely right.
Thank you. And our last question isn't a question, it's just a comment, and I think it's a good one to end on. It's from Larissa Constantino, and she says, "Thank you very much for your talk and for sharing your personal moments."
Oh, thank you.
Thank you, Dr. Xu, and I'm sure our paths will cross again, considering we're both in USDA.
Yes. Yeah, thank you for having me.
Thank you.